So the first module that we have to work on is a background in protein crystallography. Some learning objectives are how protein structures can affect crystallization, become familiar with structural biology rules that govern interactions, and how to analyze active sites to figure out how enzymes actually work. So you don't really need to know much more other than general chemistry at this point, and I suppose biochemistry would probably help too, but you really just need to know what a protein is, which is something you can learn as a freshman or upper level, even high school course. So the figure that I have here is what I call the Pac-Man model, and it's an example of how enzymes are often introduced in concept. So in this example, we have an enzyme that looks like Pac-Man and a substrate that looks like a piece of pie that fits directly into his mouth, and then when he binds, there's some sort of lysis reaction into smaller pieces of pie. And this is fine for introducing the concept of what an enzyme is, but if we have atomic level resolution structure, then we know better what the enzyme looks like, and specifically the amino acid residues that play a role in directly interacting with the substrate to catalyze the reaction. Something that's hard to understand is enzymes are catalysts, but what exactly do they do? Well, how they perform their job as a catalyst depends on their structure and the active site geometry and the amino acid residues that are involved. And if you compare different enzymes, you'll find ones that are similar, have pretty similar active sites. Some residues are exclusively conserved. Sometimes you get some variation. So a glutamine or an asparagine residue may be interchangeable between two enzymes because they more or less have the same functional group, they're just different inside. But it can lead to some interesting complications. And so that helps us understand better what the enzyme is actually doing when it catalyzes a reaction. It's stabilizing transition states, it's putting the reactive parts of the substrates together, it's facilitating bond cleavage so that the products will be stable. And then, of course, it has to bind the products weakly so they fall out. And so all of this plays a role in how they actually carry out the job of catalysis. And so when we have an actual structure, we can see directly what it is they're doing, and we don't have to guess anymore. So what's actually going on? What affects things like protein folding and the actual structure of the protein? Well, a lot of things. There's a video that's in here, very brief video that shows what actually happens as a protein's folding. And something that I think is often forgotten, because it isn't always shown this way, is that when a protein folds, it doesn't just get spit out of the ribosome as a polypeptide, it's folding as it's being made, because protein folding happens faster than translation. So as the protein's coming off the ribosome, it's interacting with the surface of the ribosome as well, and that can affect its folding. So certain parts of the protein will fold faster than others. There's intermediate shapes. And of course, this is generic. There's every single protein is going to have a different way or different mode of folding. Some require chaperone. But it is deeply complex. And so when we think about how a protein is going to fold and what affects that, there can be a lot of different things. The final protein may require post-translational modifications as well. So it's very involved. And we could say that, well, we just have AlphaFold fold everything for us and give us protein structures, but AlphaFold does a pretty good job of it, but sometimes it doesn't. And so you read through the module, I give examples where AlphaFold did a really good job folding ribonuclease, but a small sulfur transferring protein that is, of course, significantly less known. It did not do a very good job of it. So these are just examples of how if you use AI, it's fine, but sometimes you have to look at the output and make sure that it makes sense. Clearly, what it gave for the output for this protein was not correct. And so there's a lot of secondary structure that doesn't form. It doesn't seem to be very coherent and very cohesive. So this isn't particularly useful. But AlphaFold's making improvements over time, of course, but it will not tell you things like if a ligand is bound to your protein, it can't necessarily tell you if a metal is bound. It won't tell you if your protein has an interesting binding partner or anything else that could be involved in the biochemistry and ultimately what its role is inside the cell. So that is an important part of the structure, but there's more to it than simply the structure alone. So we do need to continue to do crystallography, and it is a very important skill because there's a lot of stuff that we can learn that we can't learn simply, of course, from the primary sequence alone. One of the things that plays a huge role in how proteins function is their overall charge and, of course, how they crystallize. So differences in how a protein crystallizes can depend on the pH because that affects surface electrostatics. So something that is worth considering. And one of the biggest things we do when we're trying to screen for protein crystal conditions is change the pH. Because change in pH can lead to dramatic changes in surface electrostatics. And that's largely what's causing proteins to come together to form a crystal in the first place, is how do they interact with each other? So that can that can be affected by the pH. But the actual crystallization itself is very challenging. I have here an example of a screen that I had set up on my own when I do my own crystallography screening. Once I find a condition that looks like will grow crystals, make some slight changes with different types of buffers at different pHs that ultimately lead to differences in pH, as well as different concentrations of, say, precipitant. In this case, it's polyethylene glycol 3350 or PEG 3350. And whatever else happens to be in your conditions that have to change. Sometimes there could be different amounts of salts or other types of additives that help. Often not just one condition, one specific precise condition will give crystals, but a range within a certain range of pH and precipitant concentrations. And sometimes just setting up a bunch of crystals and they sort of harvest them so that you have a lot of samples to bring to a synchrotron or a home source is the best way to go. Typically, this is handled by doing some kind of drop experiment. Either hanging drop is probably the most common, but sitting drop and sandwich drops work too. Just depends. There's some slight variation in how fast water will evaporate and concentrate the sample down and would crystallize it. So the initial module is just background in crystallography, hopefully from a perspective that's different than a typical biochemistry text would do. Certainly don't need a rehash of, well, here's a protein, but I certainly do think that there are some specific considerations that are involved in crystallography that I've covered here that should help couch the challenges that we see when dealing with crystallography.